Hi friends, welcome back to the part 2 of the presentation on culture media. So in the first part we have seen some basic things, what is the need of culture media, and especially we have seen what are the different types of media as an outline. And also we discussed the first criteria for classification. That means that was based on their consistency. And in this second part of presentation we will see the classification of media based on their composition. That will be the first thing we address, and the second thing is we will see a number of special media including enriched, selective, differential, etc. So their special applications and other things will be discussed. Okay, so let's move to the presentation. So based on composition or ingredients of the media, we have simple media, complex media, and chemically defined media. So simple media is simple in terms of composition. Sometimes we use nutrient broth and nutrient agar. We consider them as simple media as they consist of limited composition or limited components as the nutrients, right? If and also we can have examples like peptone water and other simplest media are there, which are used in microbiology laboratories. Okay, it's purely based on the composition complexity. Okay, the composition, when it is quite simple, then we use the term simple media. Now the second class is complex media, which usually contain some unknown chemical composition. That means there are the ingredient, all the components are not known, right? That means the exact composition of the media will not be available, will not be aware, but it's a nutrient component. It is definitely it's a complex nutrient material, but the exact component, that means whether it contains nitrogen, carbon, sulfur, uh, which substrate provide all those things may not be exactly known, right? So will provide all the nutrients and vitamins required for the growth, and usually there are certain things like yeast extract, animal extract, plant tissue extract, soy digest, then some milk based concentrates, peptone, even peptone, all are some ingredients which are used in complex media, right, or which makes them complex. See, the all the sources will be satisfied, like there will be energy, carbon, nitrogen, sulfur, all the requirements of the microorganisms will be provided usually or the primarily by a protein source. That's important. Usually in complex media, the energy, carbon, nitrogen, sulfur, and most of such requirements are provided primarily by proteins. That means, see, when you see that peptone, soy extract, plant tissue extract, animal tissue extract, or yeast extract, all those are rich in protein. So these proteins will provide all those, they will meet all those requirements. Okay, then vitamins and other organic growth factors will be provided by the meat extract and yeast extract, in addition to the already said sources. Then yeast extract usually considered as a good source of vitamin B or B vitamins, and also carbon and nitrogen source. Clear? And it's useful when or in which condition we use this complex media. It's widely used when the nutritional requirement of an organism is unknown. In such a situation, we cannot define a composition. That means if you have a bacteria with you, and the composition or the need of that organism in terms of their nutrients is unknown, then in that situation we will provide a very rich media. That means very complex media which contain many substances which could be used by the microorganism. right? So this media are sufficiently rich and complete to meet the nutritional requirements of many different organisms. That's important. So if the nutrient requirement of an organism is unknown, we can go for complex media as a choice, right? Again, nutrient growth is again coming under this also as an example. Then Mekong agar, tryptic soy broth, all those are different examples. Then the third class, a defined media or a synthetic media. As the term itself indicates, this is the media in which the exact chemical composition is known. So those who are preparing the media, they know what are the components and what component will provide energy source, what will be providing the carbon source, like that. So the pure chemical substances are usually preferred for the preparation. The number and the complexity of ingredients may vary from organism to organism and from medium to medium, right? Occasionally, maybe there will be certain media which use only a single component, and there will be certain other media which use a number of components, right? And definitely this will be useful when the exact chemical growth requirements are known for an organism. In that situation, we can choose a defined media rather than a complex media. You can see example, peptone water is a very widely used example for cultivation of various organisms. The second example is provided also there for the cultivation of E. coli. You can see glucose is provided. It's simply a carbon source and also could be an energy source. Then ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, etc. are there. So this is a defined media. Exact composition is known. Glucose is a non-pure chemical which has carbon, hydrogen and oxygen, and also could be used as an energy source. Then ammonium phosphate will provide ammonium and phosphorus. Then sodium chloride will provide sodium, Na plus and Cl minus, and also will maintain the osmotic balance. Then magnesium sulfate provide Mg, SO4, Mg2 plus and SO4 minus. Then sulfur and magnesium. Then potassium and phosphorus will be provided by potassium diphosphate, etc. So the exact components is known. All the composition and all the sources are known for a defined media, right? So this is the second criteria used for classification. That means simple media, complex media, and defined media or synthetic media, right? Hope it's clear. Now we are going to discuss some special media, and one of the first thing we are going to see is enriched media. It's a special media, right? And usually it's preferred for the cultivation of nutritionally exacting organism, or otherwise called fastidious organism, right? What's a fastidious organism? Fastidious organisms are the one which cannot survive in a normal media unless special nutrients are supplied in the growth medium. So this is a special organism. They do not grow when a normal nutrient media is provided. They need additional ingredients for their growth, right? It's simply like they'll grow only if the medium is little rich, or a very good nutrient medium is preferred by a fastidious organism. Okay, so there will be added ingredients to the basal composition. That's why it's called enriched. It's an enriched media, enriched by addition of additional nutrients. Fine. And usually what things are usually added to enrich it? Usually substances like blood, serum, egg, and some other growth factors are usually used to flourish the growth of fastidious organisms. Fine. And we can see a lot of examples like blood agar, chocolate agar, Leoflace agar, or LJ medium. LJ means Lowenstein Jensen medium. So the added things are also given there. In blood agar we add blood. In chocolate agar also we use blood as the added ingredient. In Leoflace we use serum, and in LJ medium we add egg, right? So there will be different organisms, they prefer different ingredients, and based on that we use the media, enriched media. And here you can see a little explanation on blood agar and chocolate agar. And both the media are enriched by blood. These are enriched media used for cultivation of fastidious organisms, and also there will be some other purposes, right? See, first see blood agar. Blood agar is enriched by addition of blood and will be definitely will be red in color. Used for the cultivation of fastidious organism and also is helpful to check the pattern of hemolysis. Hemolysis means the breakdown of the RBCs that leads to hemolysis. That means that leads to a clear zone formation. We will discuss that later in detail. At the same time, chocolate agar is also added with the blood. But the difference between the blood agar and chocolate agar is that the blood agar consists of blood which are added after cooling the medium, right? Or a fresh blood is added. At the same time, into the chocolate agar, the blood added into the medium is heat treated, or otherwise you can say the blood is added into a media which is having a particular temperature, right? So chocolate agar is enriched with heat treated blood, usually around 40 to 45, or even some little higher of 45, which turns that's why there is a brown coloration to the chocolate agar. That's why it's called chocolate agar. It's not because which contains chocolate or anything, which is because this has a color of a chocolate. That's why it's called chocolate agar, and used for the isolation of fastidious organism. Okay. Then the second special media we are discussing is enrichment media. Okay. So these are liquid media used for selective cultivation of certain organisms from a mixed population. Suppose imagine you have a sample, or maybe soil sample or water sample, and we have a number of bacteria present in that. And in that situation, and if you want to isolate a single organism, if you want to cultivate a single organism from that mixed pool, in that situation you can use media like enrichment media. And here how what the purpose is, we need to prevent the growth of certain organism, or we need to positively promote the growth of certain organism, right? And how it is meant, for that we add certain substances, or we can provide certain environment to promote the growth of the wanted organism, right? The organism which we need to cultivate will be promoted, while those which are not required are not promoted. Clear? For example, if you want to isolate a bacteria which use phenol, right? If you want to isolate a bacteria which consume phenol from a soil sample, so definitely the soil sample will contain numerous microorganisms, maybe thousands of types of organisms will be there. And from that mixed pool, if you want to isolate only those which utilize phenol, in that situation what you do is, this sample will be inoculated into a media which contain phenol, right? So this is usually liquid in form. This liquid enrichment media contain phenol as the only source of carbon and energy. So what happens when the soil organisms are inoculated over there? Only those bacteria which consume on phenol will be survived. All other organisms will perish, right? So they will be removed from the media without available nutrients. Clear? So we can see various examples, like saline type broth is used for the isolation of Salmonella and Shigella from fecal samples, and also peptone water or alkaline peptone APW for the isolation of Vibrio cholerae, right? So this is enrichment media. This is nothing but a liquid media in which there are added substances, or we are making a particular environment to promote the growth of a wanted organism alone, and the unwanted organism will not be promoted. Clear? Example, salinity broth, alkaline peptone water, etc. Now the selective media. The third special media is selective media. Is designed to suppress the growth of unwanted bacteria. In the previous one, that means enrichment one, we have seen that the wanted organisms are promoted, but in selective media the unwanted organisms will be inhibited and only the wanted organisms are allowed to grow. Right, that is what the selective, we are having a selective isolation, right, usually which is a solid media, and permits selective surface growth of the organism. Right, and how do we select this organism? And we usually prefer three conditions, or either of the three conditions will be chosen. First one is addition of an inhibitory substance to the media to inhibit the growth of unwanted organism. Right, for example, you can add antibiotics, you can add antimicrobial dyes, or even other antimicrobial chemicals which are specifically inhibiting a particular group of microorganism. You will get a clear idea when we discuss the example. Then the second one is by limiting, or the second factor how we maintain the selectivity is that by limiting an essential nutrient. Right, that means a limiting factors like growth factor, maybe a particular amino acid which is not provided in the media, so that most of the organisms are not able to survive. Right, and even otherwise you can choose a pH, an altered pH will be taken. That means if you want to isolate a bacteria which can grow at alkaline pH, then the media is the pH of the media is kept at very high range, that means at alkaline range, so that only those which can survive at that high pH will be remaining in that media. All other organisms will not survive. And the third pattern we choose is by providing a nutrient which is used specifically, that means we provide a particular substrate that will be used by the bacteria which we want to isolate. Right, for example, you can clearly understand that if you provide cellulose as the source of carbon in the media, then only those bacteria which consume cellulose will be surviving, or cellulose digesters will be present in that media after incubation. Right, all the other organisms, as they are not able to consume cellulose, will not survive in the medium. You can see examples. Now the first example is endo-agar. We have different examples for this category. Endo-agar, this is one of the class of media which has wide application. Right, so the endo-agar in which sodium sulfite and basic fusion will inhibit the growth of gram positive organism. So definitely gram negatives will be promoted to grow. That's what endo-agar. Then eosin methylene blue, or EMB agar, in which methylene blue and eosin Y inhibit gram positive organism, and again gram negatives are preferred. Then McConkie, again similar way, gram negatives are selected and positives will be inhibited by crystal violet and bile salts. Then thiamartin agar, TM agar, in which certain antibiotics are added like vancomycin, colistin, those inhibit gram positives, and nystatin will inhibit fungal growth, and trimethoprim will inhibit gram negative organisms except certain specialized group like Neisseria. So this thiamartin is used for the cultivation of Neisseria selectively. All other group of organisms will be inhibited. Right. Then finally the LJ media, Lowenstein Jensen, which inhibit the growth of gram positive and also gram negative through the activity of penicillin, nalidixic acid and malachite green, and this is used for the selective isolation of mycobacteria. Right. So these are the different media coming under the selective media category. You can see this endo-agar and all other agar media, EMP, McConkie, LJ, etc. There are other media also like TCBS, thiosulfate citrate bile salt sucrose agar, used for the selective isolation of Vibrio cholerae, and which inhibit gram positives and also gram negatives like Enterobacteriaceae due to the presence of certain things like sodium thiosulfate, sodium citrate and oxgal. Then Wilson and Blair medium for the selective isolation of Salmonella typhi, the pathogenic agent of typhoid, and which inhibit intestinal gram-negative and gram-positive organism by the presence of bismuth sulfate and brilliant green. Then another one is potassium tellurite media for the selective isolation of diphtheria bacilli. Right. So all these are different examples under selective media. Fine.